Hello, everyone, and welcome to today's webinar titled Single Cell Mapping of Human Brain Cancer Reveals Tumor-Driven Education of Tumor-Associated Leukocytes. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Fluidigm. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on that Ask a Question box located on the far left of your screen. I now present today's speaker, Dr. Burkhard Becher, Professor and Chairman at the Institute of Experimental Immunology at the University of Zurich. For a complete biography on Dr. Becher, please visit the Biography tab at the top of your screen. Dr. Becher, welcome. Please begin your presentation. Welcome, sir.
Hello, everyone. Thank you for the kind introduction. My name is Burkhard Becher, and I'm happy to be here. Being here obviously means in my home office, where many of you, I guess, also are banned for the last couple of months. But I'm happy to share some work with you that just recently saw the light of day, and it's work by Ekaterina Friebel on the single-cell mapping of human brain cancer and how we think we found out how tumors drive the education of leukocytes within the tumor microenvironment. And let me see whether I can advance the slides here. So I quickly, this rather not so pretty picture on the right is a picture of a glioblastoma, which is very the most common primary brain cancer. It's a very aggressive brain tumor. The prognosis is really, really bad. Even though it does not metastasize, it is highly invasive. And surgeons, as you can imagine, cannot remove as much tissue as they would like, because the brain is one of those tissues that is, rather non-redundant, and it needs to, so not much, and doesn't regenerate very easily. So removal is very limited, and so the patients basically get chemo and radiotherapy, which does not work. And the majority of cases, the median survival is certainly less than 2 years. And people have attempted checkpoint inhibition and immunotherapy of patients with glioblastoma, and they largely failed. And so what is the consensus is that glioblastoma multiforme or astrocytoma, WH4, we'll talk about this in a second, is what you call a cold tumor. Now, I would like to, and it sounds as if this was the initial plan that we had in the lab, but there's a reason why I'm presenting the data this way, where I start with glioblastoma specifically, and then I go back a slide and say, what is actually brain cancer? And I didn't answer that because there are different kinds of brain cancer. So let me just quickly just say this, what we see, what we call glioma, can be this diffuse astrocytoma and other different other gliomas, but the worst of those is, as I mentioned earlier, this grade 4 glioblastoma or astrocytoma, and that is the nasty, horrible, and disease with a very, very poor prognosis. But brain cancer, and so the other thing I should mention here, just for clarification, the difference between a grade 3 and a grade 4, the lower grade tumors, and the high grade glioblastoma appears to be a mutation in this in IDH1. So IDH1 wild type tumors, so the non-mutated ones, are the aggressive ones, whereas the mutated ones are actually not as aggressive and may just represent a different tumor entity altogether. But brain cancer is also, and perhaps even more common, the site of metastasis. So melanoma and carcinoma, and for instance lung cancer, do metastasize into the brain, and that is basically the start of our study. And the questions we had is there a difference between them when it comes to the tumor microenvironment? In other words, is the tumor microenvironment of brain cancer in general shaped by the particularly unique structure of the central nervous system. So by this, let's say it used to be called immune privilege of the brain, and the particular microenvironment, is that shaping the tumor microenvironment, or is actually the tumor microenvironment specific to the kind of cancer that happens to be in the brain? So it's dictating, so it's the tumor cells that dictate the behavior of the leukocyte and the tumor microenvironment compartment. And another introduction I'd like to give here is the brain is also a complicated site when it comes to the resident population of leukocyte, in particular when it comes to the macrophage. So the macrophages of the brain, the tissue resident macrophages of the brain, are almost all of them are actually so-called microglia cells, and they come from a very early embryonic precursor derived macrophage, in fact from the yolk sac. So they populate the brain early during development. And then that is self-repopulating, self-sustaining population of phagocytes that live in the brain. And when we're having this discussion, what is a tumor-associated macrophage? So in the brain, you'd have to think of the microglia as the primary tumor-associated macrophage. But at the same time, the pathogenic insult, so the change of an alteration in homeostasis in the brain, leads to the influx of inflammatory cells, such as monocyte-derived cells that we call monocyte-derived macrophages. And those cells then will be also tumor-associated macrophages. So on the one hand, you have to see an as resident population of macrophages responding to the kind of cancer or to cancer in general, to cancer cells within the brain. And on the other hand, depending on the level of leukocyte invasion, you have monocyte-derived cells and other macrophages and other phagocytes moving into the central nervous system from the outside. And the problem is, we talk about TAMs a lot, but it's a rather diffuse entity by the definition of being a tumor-associated macrophage. And it may very well be, and we assume that the resident population of macrophages and the invading population of macrophages have significantly different behavior in the context of cancer and maybe even in the context of patient outcomes. And this is a particularly problem to study, is well suited to use high-dimensional cytometry. Now, single-cell technologies is the ones I put this little slide in here just simply because I'm annoyed sometimes by people saying, oh, we did some single-cell analysis and we found this and this and this, and they usually refer to single-cell RNA sequencing technologies, which we're using in the lab as well. But I'd like to just point out that immunologists have been using single-cell technologies since a very, very long time. So the first fluorescent-based flow cytometer is actually older than I am. Not much, but it's older than me. And then first multicolor flow cytometry became basically with in the 70s, 80s, and then basically 11th color flow cytometry in 2001 was the first time somebody showed an 11 parameter flow cytometry. I don't want to, in this particular with this particular audience, I don't have to tell you the power of one additional parameter and what it does. You can see here, however, what I wanted to stress is that I believe the technology of single-cell cytometry to have a couple of features which are obviously potentially even superior to what we see with single-cell RNA sequencing, in particular when it comes to the amount of throughput that we're using. But by now, I mean, this publication is a few years old from which I stole this picture. But we have the third generation mass cytometer is on, and there is constantly more development. Again, in this setting, I don't have to explain this to the audience. But there's a technology I personally also find quite interesting, which is spectral analysis, that is now basically can be considered quite competition to mass cytometry. But let's just move to the data. This is the picture of the two investigators in our team. That is Ekaterina and Susanna, and they worked on this project together. And basically what they decided to do, and it's also because of the collaborators that we have at the University Hospital here, and at the neuro-oncology and neurosurgery department is that we had unique and privileged access to tissue that is resected from patient brains, and that we at first glance did not know what kind of brain cancer tissue we were getting here. So we would make a single cell suspension, run it through the mass cytometer, and had, in this case, 74 different markers. And here on the, you see this dimensionality reduction image. I forgot whether it's a TISNE or UMAP or what have you, but the point is what you're seeing here is that we can clearly see demarcate populations, clearly identify them. I will not share the heat map and all the details with you, but basically see that there's a rather rich environment. This is now from a composite image of all the different samples that we have analyzed. But when you then ask the question, so the first question I mentioned earlier that what we had was, is the type of cancer dictated to a microenvironment, or are they all basically dictated by the brain? And this answer was rather easy to find. This question was rather easy to answer. As you can see here on the right side, this is basically a PCA plot that clearly shows that glioma and metastasis cluster completely separately and really crisply differently. So while the glioma samples cluster in this one spot, the metastasis cluster in a different spot. And you might want to know, so a PCA that is not the most sophisticated way of analyzing, because this is a linear PCA, of analyzing nonlinear data, but this is basically driven by T cells. So in metastasis, we find this accumulation of T lymphocytes that are relatively rare in the different glioma entities. And so as a control, what we see here is we used, and throughout the presentation and all the data are relying on patients with epilepsy, mutated gliomas, wild-type gliomas or glioblastomas, and metastasis. And they're separated here in these 5 different tumor entities. It was interesting to us to see that the treatment, for instance, with antibodies to co-inhibitory molecules did not influence this clustering at all. So T cells are dominant in metastasis, and we don't see them so much, obviously, in the brain endogenous cancer, the cancer that comes from of neuronal origin, of neural origin. And epilepsy is the closest we could get to healthy brain, fresh, healthy brain from a living patient, because as you can imagine, getting tissue from, it's difficult to find donors, obviously, of brain. But epilepsy, we thought, is the closest approximation to healthy brain. And you can see here in this image that there are very few T cells in epilepsy brains. But let's just quickly go a little bit deeper into the data and look at, for instance, these so-called TAMs, the tumor-associated macrophages. And what we did, and we did this for quite a while now, both in mouse and man, to make sure that we know the difference, the phenotypic difference between a microglia, between a monocyte that is blood-derived and a monocyte-derived macrophage, we call them here. Other people call them monocyte-derived dendritic cells or monocyte-derived cells in general. Here we just call them MDMs. And we used mice in addition to what we're doing. I only show you human data, but let me assure you that we know, for instance, the expression of CD49D is exclusive to blood-derived cells. So the microglia are CD49D negative. We verified that also by histology and by fate mapping in mice, and we've done a lot of extra effort. It's all in the paper. If you're interested, you can read this. It just came out earlier this year. You can see that the monocytes and monocyte-derived macrophages are clearly different from the microglia. But now we can look at these cells in some more detail. And now that we identified this, and you can see here, again, let me just go back one slide, I apologize. You can see once again on the upper left panel, you see this TISNE plot that I showed you earlier, or UMAP plot that I showed you earlier, where I basically say now we're focusing on the phagocytes. And within the phagocytes, this here is a force-directed, this is a scaffold plot that shows you the monocyte-derived macrophages, and the microglia. And on the right side, you find the heat map. But this is a composite of all the samples, whereas here we separate the samples. We look at, for instance, what it looks like in the glioma in glioblastoma, so the wild-type IDH1 wild-type cases, melanoma and carcinoma. And you can see immediately from a distance that the mutated low-grade glioma has relatively little influx of monocyte-derived macrophages. And what we're seeing much more influx in the glioma cases. So glioma, it seems to attract monocytes to then convert into these TAMs, monocyte-derived macrophages, and that that still is dominated by microglia, and that the number of microglia or the number of monocyte-derived macrophages increases in the metastasis rather dramatically, as you can see here. And then this is the relative contribution then of microglia to the TAM population is obviously then reduced. This isn't, when we studied the microglia in some more detail, I can just show you here, this is just, again, just a snapshot of something. When we look at the CNS resident phagocytes, yet they are responding to the within the tumor microenvironment. So for instance, I don't show you epilepsy because it basically looks similar to the grade 2 mutated gliomas, whereas in the higher grade glioma, you can see already the microglia upregulating MHC class 2 and other markers and the heat map, and all the raw data are with the paper, and you can download them for your perusal if you want to. But what I thought was much more interesting is to look at the monocyte-derived macrophages. And this again, on the upper left, you can see here, we basically draw the gate on the CNS invading phagocytes. And then using this slingshot pseudotime analysis of the data, we start on the lower left. I'm sorry, now we're talking at the very middle of the image that I'm sharing with you right now. You can see we're starting from the classical monocytes, and we can see the strange trajectory. Once again, this is the composite of all the cases that we have. We see the strange trajectory. I will not go into detail about the trajectory, but instead what I want to show you and what I want to share with you here is the fact that it does look, again, when you tease the samples apart and you look at the wild-type IDH1 and the melanoma, carcinoma, metastasis, you immediately see a fundamentally different shape in the tumor macrophages in the MDMs, and there's a rather clear fractalkine receptor and CD163 expression in the monocyte-derived macrophages in, for instance, in the glioma cases, in the glioblastoma, whereas we find an upregulation of 206, 209, and 69 in these melanoma cases and in carcinoma, and there's also this increase in PD-L1. So you see this bifurcation of macrophage education that is driven by the tumor entity. So the tumor-associated macrophages of a glioma and of a melanoma and carcinoma are actually different from one another, but within the melanoma and within the glioma, for instance, they are relatively homogeneous and clearly indicating of a tumor-driven education or tumor-driven polarization of the macrophage compartment. And this is not just an interesting phenomenon, but it's actually something that is relevant also to the patient outcome. The particular signature here, in particular in the low-grade glioma, shows you that this accumulation of this particularly polarized macrophage, fractalkine receptor and 163 positive monocyte-derived macrophages, is if you do see a high prevalence of these cells in the signature of this is a TCGA database, it does actually correlate to poor patient outcome. So there's an indication that this is actually something that we call a suppressive monocyte-derived macrophage. Other people have called these entities myeloid-derived suppressor cells, a term that I wouldn't want to use here because I think it is slightly too nebulous to make sense of it. But at this point, this is just a correlation. If you have this particular signature in a patient, that does correlate with poor outcomes. And for us, it will be the next step that we're undertaking right now, is to understand the biology of these cells. Why are they actually suppressive? What do they do? And there are some indications. There's also a little bit of literature. But the advantage of the technology that I've showed here, in this case, it's mass cytometry, but also high-dimensional cytometry in general, is that you can separate these entities, study them in more detail, also in preclinical models, which is, I think, where the next step is to validate some of these findings. Now, having talked about macrophages, which is clearly also the focus of that study, there are, of course, some insights into the tumor invading lymphocytes. And I told you earlier already that we see many more TILs in the metastasis than we see in the gliomas. Here, basically, we are looking at a subset of TILs, namely the regulatory T cells. And there is this story that's been out there for the longest time that because glioblastoma is a relatively cold tumor, that we would see this accumulation of regulatory T cells, and there are numerous publications to support this. And we were a bit surprised to see that the biggest difference that we see between the samples was, in fact, that an accumulation of regulatory T cells in the metastasis cases. So those that see more invasion of leukocytes altogether actually also see more regulatory T cells going in. And that while when you really think about the data, it's actually not really surprising anymore. But we were a little bit surprised by the fact that we found relatively few Tregs in the glioma cases. There are some, if you think you can go further, and again, this is more of a resource for people to look at. If you want to go into the data, you can see and study more the changes in the resident compartment or in the resident memory T cells that we see, and we see clear changes in the response of the resident memory CD8 T cells. This is just one example of things we've studied. But the presentation here is basically just a snapshot of a rather rich data set, which is now as a resource online where you can just download things and study questions that you had and try to answer them with the data set available. There are also the possibility for us, I mean, obviously, when we look at TILs, you might as well look at natural killer cells. You can see again in the upper left corner, just that we put the heat on we gated on the NK cells and found then indeed that once again the tumor type dictates the behavior of these natural killer cells. In particular, we found this population of CD56 intermediate to bright and CD16 negative NK cells, particularly in the glioblastoma cases, and there were almost none of them in melanoma and carcinoma. And let me go further in the interest of time. So we have the impression that immature natural killer cells do accumulate in this, as immature based on their phenotype, and NK cells accumulate in the glioma, in the wild-type glioma. You can see this here. Some other information about the proliferation of the NK cell compartment, expression of 2b4, TIM3, and what have you, and proliferation is measured by Ki67 staining here, and you can see the data right there. So what is the bottom line here is, and that's something I really wanted to share, is that this high-dimensional cytometry that did capture 100% of the leukocyte landscape in the human brain. So it is not that there were any unknown entities, cells of what are they, we don't know, or immune cells or something. We could really define each cell population in quite great detail using this technology, and that with a fair number of patient samples that we had available to us to make this comparison. The CNS resident phagocytes clearly dominate in the glioma tumor microenvironment, and they seem to acquire an immunosuppressive signature. And the monocyte-derived macrophages, they accumulate more in the high-grade glioma, but clearly dominate in metastatic lesions. And the composition of also the tissue-resident T cells, and so we talk about T and NK cells, is determined by tumor origin and not by the tumor site, which, when it comes down to it, something we were completely agnostic about what the result would be. It could have very well been that the brain dictates the TME, and you just wouldn't see major differences in the leukocyte composition other than number, but we do see a fundamental change dictated by the tumor itself. And with that, I would like to just basically acknowledge the team, and I show you this picture here because at one point, I guess if I have another minute, I'll just ramble about this a little bit. I was frustrated by lab pictures that we took and that many other people are taking, or pictures of the institute, which I never thought were really fun. And I, as a student, I always did a little bit of photography to actually make a little bit of money on the side. And we decided after many, many years of not having used my camera, take out the camera equipment, get together in the lab and take this lab picture, which by now some of these are no longer in my team. Some few members have joined already. But this is basically the lab team. I acknowledge in particular the two people that ran the study, which is Ekaterina. She's right here in the very middle. But since I don't have a laser pointer, I showed Ekaterina to you earlier. And then we have a bunch of nice collaborators. And if you're interested, you can follow us on Twitter. And I wish to thank you for your attention. Thank you very much.
And thank you so much, Dr. Becher, for that informative presentation. We will now start the Q&A portion of this webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We already have some questions coming in from our audience members. Dr. Becher, for what kind of experiment do you use scRNA sequence when single-cell cytometry?
Okay, can you hear me?
Yes, we can.
Okay, very good. Yeah, I think this is a discussion we have with so many people. Always, there's this huge discussion. I told you earlier when I looked at the presentation just now, when we talk about single-cell technologies nowadays, everybody refers to single-cell RNA sequencing, which I think is just utterly wrong. Because if you look at a microscope and you look at the morphology of single cells, you're studying single cells, even within the context of a tissue or what have you. You're still a single-cell analysis ongoing here. The issue is then a question of dimensionality, or better to say, question is the number of parameters you're studying. Single-cell RNA sequencing, of course, has the highest number of parameters you can study. You can basically look at the transcriptome. If you spend the money, you can even go in quite a bit of depth in there. But I sometimes find it frustrating because you do have, for instance, if you study immune cells, I rarely see a novel set of immune cells popping out and being discovered this way, because the dimensionality threshold for immune cells with our panels for cytometry are often met quite quickly already. So there's very little need for this. Sometimes we use RNA sequencing to go in depth in certain populations. We're curious about what they're doing. We're curious about their shape or their development. Then this is a good technology for, let's say, preclinical studies. But when we look at clinical data sets and we are interested in studying large cohorts of patients, either blood or tissue, then we generally go for proteome cytometry. We go either for FACS, spectral analysis, or what I showed you here, a mixture of CyTOF and FACS.
Thank you so much, Dr. Becher. Our next audience question, when would you choose CyTOF and when spectral cytometry?
Yeah, this is the interesting thing here is that, of course, they are relatively competing technologies, and it's difficult given that forget the sponsoring right now. But the issue is we do use both technologies. I have some level of excitement about spectral cytometry because of its speed. It is so much faster than CyTOF. So the advantage of spectral and of flow maybe for so the great advantage is, for instance, you have a better dynamic range, you have more sensitivity, but then the post-acquisition analysis of fluorescence data is very, very, very demanding, very difficult to do, and fraught with lots of different problems you're facing. Whereas if the CyTOF, the data that come out of the CyTOF are basically ready to be analyzed, which is one advantage. There's very little transformation needs to happen. So I like that about CyTOF. But what I'm attracted by right now is the possibility that Spectral offers the potential for very fast analysis of huge numbers of cells. And that's where I find quite attractive.
Thank you so much. And again, I want to thank our audience for these great questions coming in. Any questions we cannot answer today will be answered via email. Our next question, Dr. Becher, how would you treat glioblastoma given that current immunotherapy modalities are failing?
So this is, of course, a great disappointment, and I've always been interested in neuroimmunology, and I've always felt that in order to treat a disease like glioblastoma, you have to have some level of inflammation. And like everybody else, I had high hopes that checkpoint blockers would make at least a little dent into it, but not even a scratch on the surface was observed. So the thing is, it seems to be that the brain is not an easy site to target, and it may very well be also because the glioblastomas are cold, and it's you need to make a change within the tumor microenvironment first, and so my thinking would be. So this is all bad about glioblastoma. There's one advantage glioblastoma has over many, many other cancers, and that is that it doesn't metastasize. So it means that if you are capable of altering your tumor microenvironment within the brain, so all these patients undergo surgery, why not add, let's say, a cytokine into the tumor bed, alert and activate and convert, if you wish, this cold environment into, even temporarily, into a hot one, and then combine that with more traditional immunotherapy approaches that drive systemic immunity, and then hoping that this combination, preclinically, that actually works really, really well. So it would be very, very interesting to do this in a clinical setting, and I think that's where the future is, and I think we need to do it soon.
Thank you so much, Dr. Becher. And we have time for one more question. This next question is actually a two-part question. Have you ever studied peripheral blood monocyte in GBM? And if so, is there any phenotype corresponding to MDM?
That's a great question. So it's a fantastic question. Unfortunately, it's not that trivial. Of all the patients that we have analyzed here, where we had access to the brain, we obviously also had the possibility to look at peripheral blood mononuclear cells. Now, in theory, PBMCs do contain a fair number of monocytes. The problem is, in order for the workflow to work, we need to, and one of the advantages, for instance, of using mass cytometry here is the capability of barcoding. So we can combine all the samples, eliminate all batch effects, eliminate all, or the majority of batch effects, and really get a really clean and crisp and nicely aligned data set. And we did the same thing simultaneously with the peripheral blood. But in order to do this simultaneously with the peripheral blood, the blood has to be frozen. The cells have to be frozen and then thawed again. And then the monocyte compartment is one of the first that seems to suffer tremendously. So while we have looked into this, we don't think that there was a good correlation on the monocyte on the few monocytes that survived the procedure to show us, to guide us, now this will become monocyte-derived macrophages that we see in the brain. In addition, I actually think that they all go into the central nervous system equally open to suggestions, if you wish, and then once they enter the brain, then educated by the tumor site and get their specialization, their transformation, their these phenotypic changes that we see occur within the central nervous system and not likely outside, I think.
Thank you very much, Dr. Becher, and thank you for your important research and your presentation today. Do you want to provide the audience with some closing remarks before we go today?
Oh, I've spoken enough. I'm good. No, thank you very much. If there's any more questions, you're welcome to just contact me and let me know. Everyone who's listening who is interested in, please look at the paper. It's a really cool resource. Check it out, look at the data, analyze them. And if you find something interesting in there, let us know and let's talk about this. It would be great.
Well, thank you very much again, sir. And I want to thank Labroots and Fluidigm for sponsoring today's event. Before we go, I'd like to thank our audience for joining us today and for their interesting questions. And questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand through July of 2021. Until next time, be safe. Have a great day. Bye-bye.